Good morning, everybody. I would like to welcome you to this lecture series on microfluidics, expanding the frontiers of medicine. We are featuring Dr. Jhalique Jane Fojas, who is in town as a Balik Scientist awardee under the Department of Science and Technology, Philippine Council for Health Research and Development Balik Scientist Program. She is being hosted by the Department of Chemical Engineering here in UP Diliman, as well as the Office of the Vice Chancellor for Research and Extension and Biotech and IPB in UP Los Baños. This is a 3-lecture series via Zoom, and we are also live streaming in Facebook through the UP Diliman Chemical Engineering Department Facebook page. So kung hindi po nakapasok sa Zoom ay pwede naman pong makita din sa Facebook. Dr. Jhalique Jane Fojas is an alumnus of UPLB, where she earned her BSCHE, and UP Diliman, where she earned MS and PhD in CHE. And she has also studied and trained at Harvard, at MIT, Oxford, and Cambridge. She holds 2 PhD degrees and several master's degrees. Her most recent doctorate was in nanoscience and nanotechnology neurophysics from Cambridge University, where her work involved the development of a brain on a chip with a microfluidic system for the study of neurons. Her first PhD degree involved drug encapsulation and drug development for anti-inflammatory proteins. She is currently enrolled at the Harvard Medical School with a view to coming back to the Philippines to train budding scientists and engineers. Today, Dr. Fojas' lecture is on revisiting organ on a chip, the future of drug discovery. So I will give now the floor to Dr. Fojas.
Hello, magandang umaga sa lahat. Just in case the internet companies fail us, brothers scam us badly today, like many other days, I took advantage of one of the many inventions of Thomas Edison and made an audio recording. It's a bit boring, but the recording, not the science. So just in case you have that, I know that it's a challenging time for all of us. So thank you for being here, to be living in such a historic year, I would say, but equally, it is an excellent opportunity for us scientists and engineers to actually adapt and ignore Darwin's pessimism on evolutionary rate. So hopefully we take on Maslow's work to heart and strive to make a difference to the lives of the people around us, particularly those who have been impacted gravely by this pandemic. And let's use science for that. Since you're all here listening to this talk, I can only assume that you're part of the fortunate 70.7% Filipinos, I believe, who have access to internet, rather intermittent access, and using this access to upgrade not just your personal computer's OS, but more so your internal OS. So if you haven't been completely booted up, grab a coffee or a chocolate to catalyze improvements on your wake up like these moments for us millennials. So we are blessed to have a working brain or cortex. That's my line of work mostly. And so let us all use that to improve the lives of our fellow countrymen and those who are less fortunate than us. I hope this lecture will inspire us just that. More than the science, it is the attitude towards its use and impact that is critical in life, especially in medical sciences, where parameter errors could mean cessation of life. Today, let's explore platforms such as organ on a chip as means to improve our odds against catalyzed death or unexpected death. So could you please move to the next slide, Sir Jumphone? Microengineering is a game-changing advancement that leads to the manufacture of microfluidic culture devices, recapitulating the microarchitecture and functions of living human organs, such as the lungs, the brain, kidney, liver, heart, skin, and even the brain. These micro devices, called organs on chips, OOCs for short, or also known as microphysiological systems, and tissue chips, offer a potential alternative to traditional animal testing. And consequently, it is a leapfrog technology that could allow high throughput drug discovery, drug testing and development at a much efficient and economical scale. Now, what is the goal of OOCs, or organs on chips? How do they work? OOCs are biomimetic systems which simulate the physiological environment we observe inside an organ, be it in humans or other animals. Therefore, OOCs should have the ability to regulate key parameters, such as concentration gradients, the fluid shear force, cell patterns, tissue or cellular boundaries, and tissue or cellular to organ interactions within the chip. In the chip, mechanical forces can be applied to mimic the physical microenvironment of living organs, such as the beating of the heart or the breathing motions inside the lungs. In many cases, we can say that they are essentially living three-dimensional cross-sections of major functional units of whole living organs. How do we design and manufacture OOCs? In principle, the process to produce different organs on chips is the same, taking into considerations the biochemical research and clinical applications. First, the design must address the properties to emulate and measure. OOCs are typically composed of a clear, flexible polymer like polythymethylsiloxane, or PDMS in short, about the size of a computer memory stick or the size of a 1 peso coin. Second, different cells must be incubated into the device. Third, cellular growth, differentiation, and function are established in order for the chip to operate like an organ. Hence, organs on a chip contain hollow microfluidic channels lined by living organ-specific cells, either from humans or other animals, interface with endothelial cell-lined artificial vasculature. So you have that vasculature there. Fourth, data is obtained through chemical and physical testing. OOCs are in general translucent, like the PDMS that you normally see in microfluidic chips, to provide a window into the inner workings of human cells in living tissues within an organ-relevant context. So this transparency of the PDMS is highly advantageous when you're looking at individual cells, for example, individual neurons, and observing the mechanism inside it under a microscope, such as super resolution microscope. Next slide, Sir Jampong. Sir Jampong, next slide po. So one critical aspect for developing OOCs is the cell type. In general, most models for OOCs are animal-based, mainly from rats and mice. However, recent advancements in tissue preservation and culture methods open new tissue sources like embryonic cells. Could you go back to the previous slide, please? Yes, thank you. So, however, recent advancements in tissue preservation and culture methods open new tissue sources like the ones that you could read here: embryonic stem cells, or ESCs, induced pluripotent stem cells, or what we call IPs, and adult stem cells, which can be differentiated and integrated into microfluidic chips. This figure is actually an excellent illustration showing the advantages, so those written in white, and limitations written in black, of these sources, including the primordial and tissue biopsies, and other cell lines. So cell lines and primary cells are more common due to their good biological response characteristics. This is what you commonly see in most biolabs. However, they do not represent normal physiological conditions, and their primary cell culture time is quite limited. Moreover, the quality of this type of cell lines are quite unstable. In contrast, stem cells are readily available and are an infinite cell source, which make them a promising technology that can be incorporated into organs on chips. Organ on chips, given its capacity to combine different cell and tissue types, present an ideal microenvironment to study molecular and cellular scale activities that underlie organ function and also mimic specific disease states, as well as identify new therapeutic targets and potential clinical interventions in vitro. So another important aspect would be the potential materials for MFE use. Next slide, please. So there are several materials that are currently being used for OoCs. In this table, you could see there, the most famous one would be the PDMS. And then you also have polymetal methacrylate, or what we call PMMA, and polycarbonate and polyimide. However, other materials have been explored worldwide, such as the family of cyclic olefin polymers that you could see in this table. Undoubtedly, though, the unique characteristics of PDMS allow the generation of the precise microchannel networks to develop in a microfluidic device. Also, its permeability has been excellent when regulating carbon dioxide and oxygen levels inside the chip. So this is why most of the biological applications using microfluidics would utilize PDMS as a material. Next slide. Organs on the chips allow recreation of therapeutically relevant interfaces, like the alveolar capillary interface, for example, your blood-brain barrier, which aid in investigating drug delivery, even drug encapsulation, as well as understanding the pathological mechanisms of diseases, and in turn, discover new therapeutics and interventions. In this schematic that you could see, it shows the multiple stages and process of drug development. So you start with basic science research, and then you move to clinical research development until you reach a point of care, a medical landscape. So if you look at this schematic, the green components represent the current and predicted use of organs on chips currently. And the blue components represent the possible and predicted utility. Many organs on chips are currently at the basic science research stage, and expanding this to medical landscape have been the current trend, targeting precision medicine and patient-specific treatments. These are particularly of great magnitude when we're dealing with rare diseases. Now let's move on and look at the various studies using microfluidic technologies for simulated organs. The basic idea in using OoCs in drug discovery and development is to provide an artificial testing subject that resembles the human body in every aspect, filling essentially the gap between extensively studied animal models and human-based clinical trials. The development of OoCs bridge the gap between in vitro and in vivo models, offering new approaches to research in medicine and pharmacology. They also open up new possibilities to investigate how environmental factors affect tissue health and physiology in individual patients and in particular diseases. One of the most momentous organs on chips would be the brain on a chip, not just because that's one of the things that I did, but this is primarily because human models are ethically not an option. So I cannot play around with your brain or anyone's brain for that matter, or simulate the pathology inside a physically healthy person. Additionally, human brain function and genetics are very different from animal models. So even during my PhD, where we work with primary hippocampal neurons of rats, that is so different. The characteristics and even the functionality are quite different from that of the human neuronal cells. Although animal models would provide a basic knowledge of the brain, animal brain diseases models remain incomparable, as I say, to human neuronal diseases. There are numerous efforts, though, directed on producing brain-on-a-chip devices to reproduce fundamental neuronal interactions and test applications with basics research focusing on the principles of brain formation and interconnectivity. And most of these applications, including brain cell function in interaction with drugs for pharmacological proposals, would try to attack degenerative diseases such as Alzheimer's, Parkinson's, to name a few. This figure from a study by Park and his colleagues, where they developed a three-dimensional brain on a chip, as you can see. This brain on a chip has an interstitial level of flow. The chip contained a concave micro wells for the formation of homogeneous neurospheroids with a uniform size, as you can observe, and 3D cytoarchitecture. They connected the osmotic pump, these are the osmotic pumps, micropump system to the outlet to provide a continuous flow of medium at the level of interstitial flow. By providing a 3D cytoarchitecture and interstitial flow, this chip approximated the microenvironment of normal and Alzheimer's disease brains, facilitating the investigation of beta amyloid effects on 3D neural tissue. So beta amyloid is one of the hypotheses that is being tested as a cause for Alzheimer's disease. Now, for the next figure, here they show the immunofluorescent images of neurospheroids of group 1. Group 1 is the static, and you have here the normal medium and the medium with your beta amyloids. So these beta amyloids have been hypothesized to cause Alzheimer's disease. And for the group 2, you have here the one with interstitial flow. And again, you have a positive control of a medium without beta amyloids and a medium with beta amyloids. The synaptic marker, which is the synapsin 2A, this one in red, and neural progenitor, or your stem cell marker, nestin, this one in green. The results of these immunofluorescent images were quantified by the intensity. So you can see here how the synapsin in those without beta amyloids are relatively higher than those with beta amyloid. So you can see that based on the intensity. Park and his colleagues determined that interstitial flow, so here, you have here the interstitial flow, actually enhances synapse formation, leading to formation of a robust neural network. These neural networks are quite important for communication between neurons and thus brain function. Moreover, they also show that interstitial flow enhances differentiation of neural progenitor cells into neurons. What does this mean? This indicates that under static conditions, the stemness of neural progenitor was maintained, whilst under dynamic conditions, you have the flow, the differentiation of neural progenitor cells into neurons was highly enhanced. So you can see that in the quantification using the intensity. More importantly, in this particular brain on a chip work, they confirmed the neurotoxicity of beta amyloids. The intensity of the red fluorescence corresponds to the dead cells, was higher in groups treated with beta amyloid, regardless of the presence of flow, as I've explained a while ago as well. Another organ on a chip that we are particularly interested in is the lung on a chip. So organs on chips also open up new possibilities to investigate gas exchange in the lungs, which is then regulated by your alveoli. So you have, where is the alveoli? So you have the alveoli here, sorry, and can be challenging to reproduce in vitro. So this whole system is quite challenging to do in vitro without using microfluidics. So microfluidics can establish extracorporeal lung models and lung pathologies through accurate fluid flow, because you can control the fluid flow inside the chips, and also you can control the gaseous exchange. Additionally, organs on chips can also be used to determine how environmental factors like cigarette smoking, for example, can affect tissue health and physiology in individual patients. You can also model a smoking machine that precisely mimics human smoking behavior and look at the impact on human lung airway functions by breathing cigarette smoke directly into the airspace of a lung airway chip. This would help in understanding the response of your alveolar sac and other exposed cells. In this lung on a chip model, a reductionist analysis of a target organ, your target organ here is the lung, identifies alveoli as your functional unit, which is essentially composed of your epithelial and endothelial cells separated by your thin interstitium. So this shows your unit. An analogous model of this system, so you can see here, the microfluidic as an analogous model, is constructed from 3 layers. You have 3 layers to bring these 2 cell types into physiological proximity. Additionally, the cells are cyclically stretched by applying vacuum. So this vacuum at the side chambers help in stretching the cells to mimic breathing-induced mechanical activity. So the result for this study in the next figure illustrates the work of Ingber and his group, which pioneered the biologically inspired design. So this is the biologically inspired design of a human breathing lung on a chip micro device. The microfabricated lung mimic device uses compartmentalized PDMS. Sorry, should have erased that. To form an alveolar capillary barrier, so you have there, using a thin, porous, flexible PDMS membrane coated with your extracellular matrix. The device reproduced respiratory motion or physiological breathing movements by applying, so here is your vacuum, to the side chambers and causing mechanical stretching of your PDMS membrane, forming the alveolar capillary barrier. Following inhalation in the living lung, you can observe that there is a contraction of the diaphragm, and this causes a reduction in interpleural pressure, or what we call PIP. This would lead to distension of the alveoli and physical stretching of the alveolar capillary interface. Now, the 3 PDMS layers are then aligned and irreversibly bonded to form 2 sets of 3 parallel microchannels separated by your PDMS membrane containing an array of your through holes. After permanent bonding, the PDMS etchant is flowed through the side of the channels, therefore producing 2 large side chambers, which is now your vacuum. And this vacuum is applied to cause the mechanical stretching, so basically simulating the breathing process. Now, this type of lung on a chip is quite advantageous when you're looking at the mechanism of how breathing actually works inside the lungs, and in simulating the different pathological conditions like like asthma, for example, wherein you could control, or hyperventilation, wherein you could mechanically control basically the movement, the distension, the stretching of your alveolar capillary barrier. Now, another organs on chip that is quite remarkable is the heart on a chip. So in this particular slide, I would be showing two of the works that have been done on heart on a chip. So as you must be quite aware of, cardiovascular diseases are the leading cause of human mortality, not just in the US, but also in the Philippines, which make the emergence of microfluidics a tremendous advancement to in vitro bionic studies of cardiac tissue. The myocardium is a major component of the heart, and the beating of the cardiomyocytes, or we call it CMs, can be used to directly assess heart pumping and drug effects. In this particular study done by Marzano and her colleagues, they developed a functional 3D cardiac microtissue by employing two separate PDMS microchambers. The CMs are positioned in the central channel to create a 3D construct, as you can see in the first figure, whilst the medium is replaced through side channels. So you have the culture medium in the two sides of the channels. The lower end of the compartment is pressurized to deform the PDMS. So you see this deformation here, and compress the 3D structure in return. So the PDMS layers are then aligned and irreversibly combined, in which the upper layers are present in the culture chamber, and the lower layers represent the drive chambers. So the key feature for this heart on a chip device is shown in the results in the next figure. So you can see the results of the chip, and yes, thank you. So these results show the ability to integrate a microfluidic platform, a simple mechanism that is able to actually convert a pressure signal into a controlled uniform uniaxial cyclic strain provided to 3D cell culture. So you have here a 3D culture, and you can see here the uniaxial cyclic strain. These figures show the functionality of rat micro cardiac. Sorry, let me just pause for a second. So this figure shows the functionality of rat microcardiac tissues. We see that the microcardiac constructs spontaneous beating monitored in four, so you have this randomly sampled 4 areas under both stimulated, so you have the stimulated and the non-stimulated or your control conditions. And this beating synchronicity was estimated by quantifying the variation of an average pixel intensity. So you can look at the pixel intensity during the contraction period, and you can plot that and show the resulting effective overlap of all the contraction peaks only in the simulated condition. So here they managed to mimic the heart using a 3D culture of cardiomyocytes. Another organ on a chip that is quite challenging to model is actually the liver on a chip. So the liver constitutes a series of complex hepatic lobules. It's really complex because it's composed of different types of cells that confer multicellular functional communication responsible for human body's major site of drug and toxin metabolism. So that's the liver for us. This multicellularity and maintenance of the physiology of hepatocytes over an extended time period make mimicking the liver in vitro a great challenge. The typical co-culture system, so if you work in a biolab, when you do co-culture system, it hardly simulates the complexity of multicellular systems like that of the liver. because different types of cells are actually mixed and seeded randomly in co-culture. So this is where the microfluidic technology would come handy. So when the co-culture, the typical co-culture method, fails to form the complex cellular architecture or manipulate cell-to-cell interaction, this could be done inside the chip by doing cell patterning. This recent development on microfabrication has provided a promising approach to establish microscale functional liver constructs on a chip, which I'll show you in the next slide. So this particular figure was taken from the works of Thaw and his colleagues. They designed a liver-based system of a liver on a chip and a matrix-dependent 3D culture. The top photo, this one, showed a liver cell culture channel of the multiplex cell culture chip. And the cross-sectional is given in the figure B. So this is your cross-sectional view of the assembled device, shows that hepatocytes in collagen gel are introduced and cultured in the bottom layer. So you have here the bottom layer. And growth medium is introduced through the top layer. At the bottom of this figure, this one, is one of its applications in which you can see the schematic of a HepG2 laden decellularized liver matrix with gelatin methacryloyl in a microfluidic device. So if you're not familiar with HepG2 is actually a liver cancer cell line. It's a human hepatoma cell line, which is often used for insulin-dependent pathway investigation. Here you also have a hydrogel precursors were injected into the microfluidic device using a pipette and photopolymerized by UV exposure to form your HepG2 laden gelatin methacryloyl for subsequent drug screening. So what they did is, because livers is actually the one responsible for major drug and toxic metabolism, liver on a chip could be used to screen different drugs and different toxins and evaluate how the system would respond to this foreign compounds or sometimes foreign proteins. Another organ on a chip that is of interest is the kidney. So kidney on a chip is responsible for the maintenance of osmotic pressure, drug excretion, where you have filtration, reabsorption taking place in the nephrons that consists of your glomerulus, renal capsule, and renal tubule. In most renal pathologies, the irreversible loss of renal filtration would lead to toxicity. In this particular chip, produced at Ingber's lab, microfluidic was used to simulate the fluid environment that support tubular cell growth and provide a porous membrane. So you would have a porous membrane support for the maintenance of cell polarity, highlighting the need for drug screening systems. More specifically, this design of kidney on a chip recapitulated the structure and function of the kidney glomerular capillary wall. The top figure shows the schematic representation of your glomerular capillary wall. Running out of voice, sorry. So you have here the podotype and the endothelial cell layer separated by your GBM or your glomerular basement membrane to form capillary and urinary compartments. And so the top right, which is the chip, shows a photograph of the microfluidic chip or device that they engineered out of PDMS to simulate this system. Below, you can see the schematic representation of the microfluidic kidney glomerulus chip with microchannels. So you have here microchannels replicating the urinary and capillary compartments of your glomerulus. The GBM is mimicked by using a porous and flexible PDMS membrane functionalized with the protein laminin. Cyclic mechanical strain was applied to cell layers by stretching. So if you stretch the PDMS using a vacuum, you can basically simulate your cyclic mechanical strain. Now, in the next slide, you can see more details of how this device was developed. So you can see here how the sandwich assembly of the PDMS channel, porous membrane, and another PDMS reservoir, and the channel can actually replicate the urinary cavity and the capillary lumen of your glomerulus. The other figure at the bottom are images, fluorescence microscopy images of the human kidney glomerulus chip established using your eyepiece, so your eyepiece cell, one of the cell sources that I've discussed a while ago, which was derived podocytes and primary human glomerular endothelial cells. So you also have here the cross-sectional view of the 3D reconstructed confocal images of your human glomerulus chip, showing the IP cell-derived podocytes and endothelial cells in their respective layers after differentiation and co-culture on opposing sides of your PDMS, flexible PDMS, which is coated by extracellular matrix as well. So this basically shows that you can replicate your GBM. Then you also have the gut on a chip, or your intestine on a chip. Another critical human body function is, of course, absorption in the gut. So nutrients and oral drugs have to be transversed in the small intestines to enter the bloodstream. So for those of you who have had breakfast, I haven't. So this is very significant because it shows you that you could actually replicate the villi and your intestine inside the chip and observe how fast the absorption of certain nutrients or proteins in the system. So the villi are actually the keys to absorption. And this is also the main parameter that you have to consider when you are properly simulating intestines in the chip. So their morphology must be maintained. Imura and his colleagues, so this is a work of Imura and in his lab, developed chips to simulate the intestinal system using a simple glass-lined permeable membrane and PDMS sheets containing channels of pure CACO2 cells cultured on the chips. So additionally, gut on a chip can also be used to culture a living microbiome for extended times in direct contact with living human intestinal cells. That could enable insights into how these microbiomes influence health and disease. So if you've heard of, the poop transplant, so gut on a chip could also be a means of learning what is a healthy microbiome inside the gut. Here you can also see in the illustration of the bionic device produced by, I think this is a work by Kim and his colleagues, with images of the device composed of mainly transparent PDMS elastomers. And you have here the cross-sectional view of the channels and square illustrations showing a top view of the porous membrane. So you have the porous membrane. You also have here the schematic of the intestinal monolayers and the phase contrast image, which indicate that you could quantify pressure if you opt to apply pressure in these channels. And so in the next figure, of the gut on a chip, the same group actually designed an MFD, or a microfluidic device, that mimics the dynamic mechanical microenvironment of the gut. It supports perfusion-based long-term cell culture with microbial symbionts and enable analysis of intestinal epithelial barrier functions in vitro. So they accomplish all this by incorporating two layers of closely opposed microfluidic channels separated by a porous membrane coated with your extracellular matrix and lined by human cathode-2 intestinal epithelial cells. So you can observe from the contrast image the morphology of your epithelial cells after 21 days of culturing, with or without microfluidic flow. So they tested ones with microfluidic flow and that of without, and also the application of your cyclic mechanical strain. The schematics of the system layout are shown on the left side. So this is basically just the schematic, while the fluorescence view is here. Show the distribution of the tight junction of your proteins. So here they use occludin, I believe, in the epithelial monolayers. And the confocal fluorescence views basically show a vertical cross-section of your epithelium, highlighting cell shape and polarity. So you have nuclei in blue and your F-actin in green. The regular array of small white circles that you can see here are pores, actually, the ones that I've shown a while ago, wherein they have like a porous membrane. While the dashed line that you can see here indicates the anchoring substrate. So you can see that there are layers in there. Now, after discussing all these 6 major types of organ chips, what shall we do with all of this? What is the benefit of actually simulating or emulating physiological organs inside the chip? What can we do with these different organs on the chips? Can we actually put them all together and produce a human chip? And this is essentially what has been done by recent laboratories in Boston and in California, wherein they put together different organs on the chips. They've started with coupling heart with lung on a chip, and then they started also doing kidney and liver. And some of them have started working with even 5 organ system to explore how complex the human system is. Because as you can see, single organ chips fail to actually fully reflect the complexity, functional changes, and integrity of the entire organ function. And therefore, constructs consisting of a multi-organ on a chips, or a 4-organ system, or a 7-organ system, otherwise we refer this as human on a chip, where multiple organs are connected like an array of physiological pathways, could give us a better understanding of how this particular organs would actually communicate with another organs inside a microfluidic device. So multi-organ on a plate systems like this shown in the figure or illustrated in the figure would give us a better understanding of how complex the system is, and how organs on the chips could actually give us a better, even a fundamental understanding of certain pathologies or certain physiological functions. So if you want to hear more about the multi-organ system or the human on a chip, or how this could pertain in drug development, drug discovery, and disease modeling, I will be giving a lecture on this in the next series, I believe, next week. So now I end my talk and I'm open to questions. I'm running out of voice. So just in case you hear some water dripping, that's me getting some water.
So thank you, Dr. Jal, for that inspiring and jaw-dropping and very informative lecture. As research and development on microfluidic applications to drug discovery proceeds toward precise and patient-specific treatment. We now open the floor for questions. I will be reading. There are already 3 questions in the Q&A box. The first one is from Jopet Ramis. Hello, Jopet. Let me read the question. In the microfluidic drug screening platform, we have been having problems with reproducibility with patient-specific iPSC-derived cellular organoids. Seemingly, it might be related on the WNT signaling. Have you looked into the WNT heterogeneity across channels of the microfluidic system on its effects towards cytotoxic or therapeutic drug response?
Sorry, ma'am. Am I on mute?
You're on. We can hear you.
Okay. So I particularly did not work with WNT signaling pathways, but the problem with microfluidics in general, when it comes to IPs and reproducibility, varies greatly based on several parameters. So it depends on if you're doing, so certain, a perfect example is just the flow. So flow with movements would change, and changes in flow would change the response of some of the cells. So the idea is to actually be able to control as many parameters as you could inside the chip. So it could be that the deviations or the non-reproducibility when you're dealing with WNT signaling pathway is not chemical, but rather could be mechanical or physical differences in the chips that you're using.
Thank you for that answer, Dr. Jal. I wonder if Jopet has a follow-up, but he's not raising his hand. So yeah, yes, thank you, Dao. So I will proceed to the next question. I have noticed, this is from an anonymous attendee, I have noticed that most, if not all of the chips have a porous membrane as its component. How are these porous membranes synthesized or made?
So you can have different materials for your porous membrane. So you can use polyimide. So polyimide, if you look at it under the microscope, they generally have nanopore size. So that could be used as a membrane. You could even use nitrocellulose. So your nitrocellulose, like your typical filters, is also a sieve and could be used as a membrane. Or you could manufacture using your PDMS. So your PDMS, you can essentially puncture that, or you could produce a mold that has, let's say, 1 micrometer of protruding pillars. So essentially you would produce a membrane by getting the negative of that mold. So there are various ways of producing your porous materials, and it depends mostly on the applications or the type of organs on the chips, because you not only want to mimic the porosity, but you also want to mimic the biological and biochemical properties of the membrane that you're simulating.
Thank you for that answer. And there's another question from another anonymous attendee. How feasible is the implementation of this in the Philippine setting?
So I think my internet connection is a bit breaking, but let me repeat the question just in case I missed the last part. But you're asking about the feasibility of doing microfluidics in the Philippines.
Yes, that's right.
Is that correct? Yes. Yeah. So microfluidics actually is one of the most efficient and economical methodologies that we could apply in the Philippines for drug discovery, drug development, and drug screening. The reason why I say that it's efficient and economical is because you're dealing with nanoliters and microliters of reagents. So you save a lot of components by using microfluidics. When it comes to manufacturing, there are several methods of producing microfluidics. And if you're dealing with sizes like 100 microns or 20 microns, you can even use some of the 3D printers to do this. But if you're dealing with nano-size microfluidics, you can do direct laser writing. And I believe that we have enough physicists in this country to establish or to even create our single photon microscope that could essentially write the geometry of the microfluidic device that we're looking into. Also, there is this called paper microfluidics, which I will discuss actually in supposedly the first lecture, which is plant on a chip, wherein you could actually do a paper microfluidics. It's very economical. But of course, the challenge is the reproducibility. But it is a technical challenge, and I believe Filipinos are hardworking. So once we optimize and create a methodology that we could reproduce, it's just a matter of doing it again and again until we perfect the system. But I think that microfluidics is highly feasible in the Philippines. If not, I think it is advantageous to do it in the Philippines.
Thank you, Jal. There's another question from Al Serafica. Hello, Al. For our Tuklas Lunas research, which chip do you think is already useful and applicable?
Sorry, I'm not sure what particular research of Tuklas Lunas.
I think it's about
Okay.
Discovering lunas from our plants.
Oh, it's a drug screening po, ma'am.
Yeah, drug screening siya.
So if you're referring to drug screening, so drug screening has several stages. So microfluidics could be used for drug extraction or your active component extraction. So you can have plant on a chip, for example, and then you can have microchips that could be used for assays. So you can have several chips lined up wherein you can test different components from, let's say, a plant extracted or different plants extracts being screened all at the same time. So these things could be easily done with microfluidics. So if you're looking at basically extracts from various plants, microfluidic is a system that you could use to do high throughput screening, wherein you could test several active compounds all at the same time in one chip.
Thank you for that, Jal. We have 3 more questions, and I think you can answer all of these in the following few minutes left. From Salvador Eugenio Tawili, how would immune cells, neurons, and or lymphatics be added to make the chips more closely mimic in vivo scenarios?
So you have different cells here. So you're looking at immune cells and neuronal cells. Am I correct? Sorry, ma'am. Hindi ko narinig masyado yung questions.
Immune cells, neurons,
immune cells,
lymphatics.
Oh, immune cells, neurons, and lymphatics. So the complexity of, are you dealing with brain on a chip? I would be guessing that you're more focused on dealing with brain on a chip. So most of the recent works with brain on a chip basically just simulate the complex neuronal networks inside, let's say, a cortex of a rat. So the immune cells could be added through your media. So here you can have the blood, you can model the blood-brain barrier such that only particular system could actually diffuse towards your neurons in the chamber. In fact, one of the things that I did when I was in Cambridge on brain on a chip is that we produce neuronal networks wherein the media is actually a simulated components of the blood. So you could do that. You could facilitate using cell media. Or I think in the long run, we would be able to do an actual human blood as media inside your chip. But of course, it depends so much on the mechanical properties. The blood is so viscous, so there is that physical strain in pumping blood inside microfluidic chip if the channel is as small as a capillary, for example.
Okay, thank you for that. There's one from Ricardo Jose Guerrero. Are they simpler to fabricate yes, yet useful OOC devices given the lack of advanced research microfabs locally. So meron daw bang simpler to fabricate OOCs?
So organs, microfluidics in general, could be fabricated in several ways. And one of the I would say the simplest way is basically to create a mold out of 3D printing, for example, in the Philippines. But the problem with that is the resolution. So the resolution of 3D printers are very low, which means that the structural size are very large. So if you're simulating, for example, blood-brain barrier, you would need to manufacture in the nano level. And that could only be done by direct laser writer. As I mentioned a while ago, this technology could be developed in the Philippines. So it's basically as simple as building a microscope that could laser out the geometry of the device that you're using. And also, with this particular question, it depends solely on the geometry and what you're looking at, what sort of microfluidics. Because if you're looking at simple transport phenomena, microfluidics, where you're looking at the transportation of a protein from one channel to another, you could basically produce that in using 3D printer. Or you can even cut it out in an acetate and produce that. It depends on the size of your protein. So this question varies so much depending on the application, depending on the geometry of what you're looking at, and depending on what organ are you actually simulating.
Oh, well, thank you. May tanong din dito, affordable daw ba sa many Filipinos itong ganitong technology?
Affordable, I would say yes, because as far as I know, I've seen several labs in UP. So if we're capable of buying an NMR, we are capable of making a microfabrication or a microfluidics lab. So definitely it is affordable. So all we need to do is to establish at least one microfluidics lab well equipped in the Philippines. And yeah, it could be mass produced.
Thank you. There's one question from Nathaniel Dugos, Dr. Dugos from De La Salle. Can we use biological ECM scaffolds as platform for OOCs?
Yes, yeah. The extracellular matrix scaffolds could be used for OOCs. And if you look at the table that I presented a while ago, you'll also see several materials that have been currently being explored by different researchers around the globe as a material for microfluidics.
Okay, thanks. And probably one of the may dalawa pa pero marami pang dumadagdag. Maybe, but it's 10:59. So probably we have room for one more. Microfluidic systems from VJ Ordilio require sophisticated equipment as well as clean environment for fabrication and operation. How long from now do you see Philippines engaging to this kind of research and technology?
Well, we're pushing for that. Dr. De Leon and another professor also from UPLB, we're looking, Professor Butch, we're looking into establishing a plant on a chip microfabrication lab in UP Las Bañas and an organ on a chip microfabrication lab in UP Diliman. So yes, so hopefully if the DOST and DOH have money, it should be fairly easy, but given the logistical nightmares and yeah. So hopefully it's, to be honest with you, you could produce a microfabrication lab at your own house. If you know, if you're in the US, wherein you could acquire reagents and stuff quite easily, it's highly doable. But right now, let's see with this pandemic. Hopefully we could produce one by next year.
Yeah. Maybe one last kasi anonymous to, so I cannot I don't know how to back to it.
Ma'am.
But do you bond the substrate to another substrate, PDMS to PDMS, and how do you deposit the cells in the porous membrane? Yes, Brian will be speaking later.
Okay.
Oh, so with the PDMS to PDMS, you can use plasma device. So plasma exposure would actually help with bonding PDMS to PDMS. But as a matter of fact, you could actually ban PDMS with PDMS without any treatment at all.
Okay, thank you. Brian, you had something to say?
Actually,
Dr.
Sir
Brian
Tito
Alman.
Oliga has a question.
Yeah, he does.
Uh,
Sir Tito
you
has three questions.
Yeah, maybe I can just mention, no, maybe John, you can answer this. Parang, how is research funding done? I mean, how do you fund this kind of work? Do we have industry players who actually commission to do the research? Like maybe the way how it's being done in Harvard, at least in Boston. Yeah, so do industry players, try to move this from research going to a market kind of endeavor as well? Do they support that kind of push for microfluidics technology?
Yeah, this is what's good with Boston area, the Harvard MIT area have a lot of startups. And what we do is that we get NIH funding to do basic research. And when it comes to actual products, we can have tie up with industries to market it or to boost more funding. And I believe that we could do the same in the Philippines because we really need the push from industry, from private companies to fund researches in biomedical sciences, such as drug screening, for example, using microfluidics. So this is something that hopefully UP and other state universities would look into, engaging with private companies.
Okay. And maybe the last one from Sir Tito. The work might involve heavy computational, work. Um, how much of, say, a computing facility, how much in terms of a server, do you think, what's the resources needed in case you wanted to do preliminary, let's say, modeling for this microfluidic system?
So what I'm using at the moment for finite element methods and
Mm-hmm.
finite difference method is COMSOL multiphysics.
Mm-hmm.
And what I have right now is actually a MacBook Pro. I
Mm-hmm.
completely forgot the specs, but it's the one that was produced way back 2017.
Okay.
And you could fairly do a good simulation using that. And at the same time, of course, you need AutoCAD as well. But yes, um, I think it will only be heavy in terms of computing when you're dealing with 3D, with several structures, complex structures of microfluidic systems. But the
Okay.
cluster should be
Uh,
fine. The UP cluster
okay, you should
should
be
be
able to
fine
do
with
it.
COMSOL.
Uh,
Yes, COMSOL and MATLAB.
all right. Thank you, Jal. There was a question earlier, but maybe I can just, we will have, just to verify, we will be sending out recordings of this meeting. And would it also be okay to share to our audience the copy of the slides?
Sorry, Brian, I didn't hear that well.
So would it be okay? We will be also sharing copy of the PDF
Oh, sure.
slides to us.
Yes.
All
Yeah.
right, and the
Yeah,
recording.
that's fine with me. Yes. Yeah.
Okay, thank you, Jal. All right, we
Thank you,
already
Brian.
thank you very much. Already couple of minutes past 11. Again, we are very thankful for our participants, our attendees this morning. And also, again, thank you very much, Dr. Fojas, for introducing, a fresh perspective on what engineering, science, medicine can do if you go on the crossroads of these three fields, that you can really do a lot. You can really solve a lot of problems, and often it would require an interdisciplinary, a multidisciplinary team to tackle complex issues. Also, again, this is just the first part of this three-part lecture series. We will have a second one on Monday and another one next Monday. So please do register so that you can also attend, and you will be provided materials and after each talk as well. This particular webinar was made possible through the support of the Philippine Council for Health Research and Development under the Department of Science and Technology, University of the Philippines Los Baños, the University of the Philippines Diliman, and we are currently supported technically by the University Computer Center of the University of the Philippines Diliman. Um, um, and then again, for those who have attended, you please feel free to share the invitation to other colleagues, friends who might be interested, who just wanted to know something about microfluidics. And we will not be holding you up anymore. Thank you very much. Hope to see you all next Monday and the next Monday as well. And enjoy the rest of the morning. Maraming salamat po sa lahat ng um, umattend at kung sakali po na hindi nyo po napanood, meron pong recording at ito po ay ipapadala sa lahat po na nakapag-register sa ating maybe wait for a couple of days pero ipapadala po ito sa lahat ng naka-register. Maraming salamat po ulit. Enjoy the rest of the morning. Enjoy first day of the week, Monday. Maraming salamat po muli.
Okay, thank you everyone, and that ends our webinar for today. Bye, thank
Thanks,
you.
ma'am.
Thanks, Jal.
Thank you, ma'am.
Yes, yes.